All right, so we're going to take a more specific look at some of the fungal phyla. And so there's kind of two big categories, the early diverging fungi and the later diverging fungi. And this has been supported by a lot of molecular evidence. And I'll just give a warning that fungal categorization and taxonomy has been changing a lot lately, by a lot, every year or two. So be aware that this information is always changing and the groupings have been changing a lot. And so we'll take a look at what the groupings are right now based on the information that we have. So the oldest group of fungi is believed to be Cryptomycota. And one of the things that's unusual about Cryptomycota compared to all the other fungi is that it does not have a cell wall made of chitin. And so remember, we sort of said, oh, well, all fungi have a chitin cell wall, but actually the Cryptomycota don't have that fungal trait. They're single-celled organisms, so there's no multicellular forms. They do live in water and soil. Their sort of claim to fame, if you will, is sort of negative. It's Microsporidia is a member of this group, and Nosema is an example, and it's a really terrible bee parasite that makes the bees really sick and unable to raise their young. So that's one you may have heard of. The chytrids are another group of early diverging fungi. They do have a rigid cell wall made of chitin. They also live in water or soil. Generally, they are also single spherical cells. Most of them are decomposers. There are some parasite or pathogen examples. And again, one that has been in the news before is Bactrochytrium, which has been found in declining frog populations. So amphibian populations seem to be really affected by climate change, and we think that one of the things that is contributing to that is this fungal growth. It's like the fungus can grow better as the temperatures warm. And so that's a big concern for amphibian populations around the globe. Okay, so I want to just take a second and remind you of the sexual life cycle of some fungi. So some of the other fungi that we're going to look at, the sexual life cycle of the fungi is distinct from other fungi. And so this becomes one of the ways that we distinguish the groups of fungi from each other is based on their reproductive structures. So remember that for some fungi, they can asexually reproduce or sexually reproduce. If they asexually reproduce, they use mitosis only. Let me write that there. To create a spore-producing structure and yield spores. The spores are not unique from each other because they're produced just through mitosis without contribution from two parents. Whereas if the sexual reproductive cycle occurs, there's a fusion between two mating types of their cytoplasm. The nuclei don't fuse. Instead, they create a fruiting body that is heterokaryotic, so there's multiple nuclei present. And then at some point within the fruiting body, usually in a specific area, there's fusion of the nuclei. And then that diploid nuclei then goes through meiosis and produces haploid spores. And then those spores, the ones produced sexually, are unique from the parent. So I reminded you of this because we're going to talk about the bread molds now, and the bread molds do carry out sexual reproduction. The most well-known example of this family of generally decomposers is the black bread mold, which there's a picture of here, and you've probably seen this at some point in your life in your kitchen because it's really common. It carries out asexual reproduction and can be quite productive at asexual reproduction, 100,000 spores per sporangia. It doesn't require a mate, so if there's a piece of bread with just one mycelium on it, it will be able to reproduce and spread to other pieces of bread. And again, you may have experience with this in your kitchen. However, they can also carry out sexual reproduction that will involve the fusing of mating types and the creation of special structures to release those spores. And so you can see some language here that's going to relate to the name of the group. So these are also called zygomycota. So they're going to actually fuse and produce something called a zygosporangium. The zygosporangium is where you're going to have the multinucleate structure, the heterokaryotic tissue. And then those will eventually pair off into the diploid nuclei and undergo meiosis to produce haploid spores. So we'll take a closer look at that. First, let's look at the asexual life cycle. So if you have a single hypha, it's going to be triggered to reproduce even if there's no mate. So using mitosis, all it does is grow these special structures, the asexual sporangia, and then through mitosis, they release spores off of this. So this will eventually have spores break off of these little structures. And these can be very prominent, like these are fuzzy, sticking off the red tissue as well. And then the spores are a way for the fungus to spread. And this is why you may have heard that mold spores are everywhere. It's because fungi are reproducing like this all the time. They can carry out asexual reproduction without a partner. Okay, so now we'll take a look at the sexual life cycle. So notice that we have haploid and diploid and heterokaryotic stage because there's a unifying event or a union between two mating types. So let's go here. So here we have our fusion of two hypha. So notice the red and the blue representing different mating types. And so these tips have multiple nuclei in them. So they fuse and form this zygosporangium that has multiple nuclei floating around in it. Okay, and it's within this zygosporangium that the nuclei are going to pair off. So a red one and a blue one are going to pair off and create a diploid zygospore. So fertilization is going to mean these two nuclei combining. And that's going to give us a diploid nucleus. And then that diploid nucleus is going to go through meiosis. And then this one's got a little bit of a, oh, never mind, sorry. Meiosis is going to produce the spores, which are haploid. And then those haploid spores can go out and make new mycelia. Okay, so again, basically like our regular structure, we're just adding on these terms zygosporangium and zygospore. Okay, so the fusion of the cytoplasm results in a zygosporangium, fusion of the nuclei produce the zygospore, and then it's the zygospore that goes through meiosis and produces the haploid spores that become new offspring. Okay, and so that's our 3 early diverging fungal phyla.